Swayam Prabha. Digital India. Educated India.
Hello everyone and welcome to the course Developmental Biology. I am Dr. Priya Goyal, Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. In today's lecture, we will discuss embryonic induction and also the fate of germ layers. This is the syllabus of developmental biology that we are following. We are done with Unit 1 Introduction. We are continuing with Unit 2, which is about early embryonic development. In this, we have discussed gametogenesis, fertilization, polyspermy, cleavage, types of blastula, fate maps, early development of frog and chick up to gastrulation. In today's lecture, we will discuss the topics of embryonic induction and organizers, and also we will take up the first topic of Unit 3, that is the fate of germ layers. We will study about the embryonic induction and organizers. We will also study various experiments done by Hans Spemann and Hild Mangold in the field of embryonic induction. We will also discuss the experiments done by Peter Newcoop and Osamu Nakamura. We'll come across important terms like Newcoop center and Spemann organizer or the primary organizer. We'll discuss the molecular basis of Spemann's organizer and regional specificity of neural induction. Then we'll take up the types of embryonic induction and organizers, and we'll discuss the fate of embryonic germ layers. Let us begin with the first part of this lecture, that is about embryonic induction and organizers. Gastrulation lays down the formation of three germ layers, that is the ectoderm, mesoderm, and endoderm. Now these three germ layers have to form certain organs, which is known as the process of organogenesis. During organogenesis, embryonic induction is an important phenomenon. So embryonic induction is the induction of embryonic cells to develop and differentiate into organs in the presence of other cells. Here we have two major tissues, the inducer and the responder. Inducer or the inductor is the embryonic tissue which exerts the inductive effect by producing signals in the form of paracrine factors. These paracrine factors are secreted into extracellular space to induce the neighboring cells. While the responder is the cell or tissue which respond to the inductive effects of this inducer. And competence is the ability of a responder tissue to respond to the inductive effect of the inducer. Evocators are the chemical substances emitted by an inductor tissue. The classic example to understand the concept of induction and competence is the building of vertebrate eye. In the initiation of formation of vertebrate eyes, paired regions in the brain bulge out and approach the surface ectoderm of the head region. The head ectoderm is competent to respond to the paracrine factors which are secreted by these brain bulges. Now these brain bulges are the prospective optic vesicles, and the head ectoderm is induced by them to form the lens of the eye. In return, the prospective lens cells also secrete certain paracrine factors which instruct these optic vesicles to form retina. So the vertebrate eye is formed by reciprocal paracrine interactions between these two regions, the prospective lens and the prospective optic vesicle region. Now here, the head ectoderm is the only region which is capable of responding to the inductive effect of the optic vesicle. As we can see, this is the complete head region or the ectoderm of the head region. And then first it is written that the normal induction of lens happens by the optic vesicle. However, if the optic vesicle is placed under the trunk tissue, it cannot induce the trunk ectoderm to form the lens. Also, if the optic vesicle is removed, there is no lens formation by the surface ectoderm of the head. Also, if any other tissue is placed under the head ectoderm other than the prospective optic vesicle or the brain bulges, then also no induction of the surface head ectoderm to form lens can occur. So this is a classic example in which we know that there is an equal importance of inducer as well as the competent tissue or the responder. There have been certain experiments by various researchers in the field of embryonic induction, in which predominantly there is role of Hans Spemann. His main focus had been on autonomous specifications versus inductive interactions. In 1903, Spemann performed an experiment demonstrating the nuclear equivalence in newt Triturus taeniatus cleavage. So shortly after the fertilization happened in newt egg, Spemann ligated the newt zygote with an infant's hair in the plane of the first cleavage. Then he partially constricted the egg, causing all the nuclear divisions to happen on one side of the constriction only. Eventually, at 16 cells pH, one nucleus escaped the ligature and came into the non-nucleated side. Now cleavage began on this side also, whereupon Spemann tightened the ligature until the two halves were completely separated. As a result, after 14 days, twin larvae developed. However, the first was slightly more advanced than the other. Thus, he concluded that the early amphibian blastomeres are genetically identical, that is, they have identical nuclei, and that each cell is capable of giving rise to an entire organism. Another experiment done by Spemann was about demonstration of asymmetry in the amphibian egg. Spemann performed a similar experiment, but in this case, the constriction was longitudinal. In one set of experiments, the constriction divided the gray crescent area into two equal halves. Each blastomere developed into normal larva. In another experiment, the longitudinal constriction was perpendicular to the plane of first cleavage, which separated the dorsal and ventral regions instead of the left and right side. So normal embryo developed from the dorsal region, which was having the gray crescent, while the other part developed into simply an unorganized mass of cells, which was known as bostick, meaning belly base. Dorsal structures like neural tissue, notochord, somites did not develop in this bostick. So this concluded that the gray crescent area contains cells which are committed to the initiation of gastrulation. In another experiment done in 1918, Spemann performed some transplantation experiments in early and late gastrula of two species of newts which were different in their pigmentation. He took the non-pigmented Triturus cristatus and dark-pigmented Triturus taeniatus. He took these two differently colored embryos so that he can identify the host and donor tissues on the basis of color after the transplantation experiments. So in this experiment, prospective neural ectodermal cells of an early gastrula of one species, when transplanted into the prospective epidermal region of the other gastrula of different species, the transplanted cells gave rise to the epidermal structures only. While when the similar experiment was performed on embryo at the late gastrula stage, the transplanted neural tissue developed into neural structures only, and prospective epidermis formed skin irrespective of the location wherever these tissues were transplanted. So from this experiment, he made the conclusion that the cells of an early gastrula were uncommitted, but the fates of late gastrula cells were determined. Some classic transplantation experiments have been done by Hans Spemann and his doctoral student Hild Mangold. In 1924, this experiment was done by Spemann and Mangold, which demonstrated the organization of secondary axis by dorsal blastopore lip tissue. In this experiment between Triturus taeniatus and Triturus cristatus, the dorsal blastopore lip was explanted from Triturus taeniatus and it was implanted into the host tissue, Triturus cristatus, into the region of the ventral side, which was supposed to become belly region. However, it was seen that the dorsal blastopore lip continued to form the structures which it was supposed to do as if it was present in its original location in the Triturus taeniatus. This pigmented donor tissue underwent invagination and it entered the gastrula through this secondary invagination in the host embryo. Now, the pigmented donor tissue continued to undergo self-differentiation and it formed cauda mesoderm and all the other mesodermal structures that normally develop from any dorsal lip. Eventually, some induced secondary structures could be seen in the host embryo. And here it was even seen that somites were formed from both the donor and the host tissues. Neural tube was formed by both the donor and the host tissues. The secondary embryo was beginning to be formed and ultimately a complete secondary embryo was formed face to face conjoined with its host embryo. Similar experiments have also been done to produce secondary embryos in other amphibian species, in Xenopus also. So Spemann and Mangold derived from their experiment that there was only one tissue in the gastrula which was autonomously derived, which had the capacity to undergo self-differentiation, and that tissue was the dorsal blastopore lip cells. So they called this dorsal blastopore lip cells and their derivatives, that is the notochord and head endomesoderm, as organizer tissues. Now this dorsal blastopore lip induced the host ventral tissues to change their fates so as to form a neural tube and dorsal mesodermal tissue, that is the somites and pronephric tubules. They also organized the host and donor tissues into a secondary embryo with a clear embryonic axis, that is the anterior posterior and the dorsal ventral axis. So here the concept of organizer came into being. They also found out that during normal development, these dorsal lip cells organize the dorsal ectoderm and transform it to form a neural tube. And they also transform the flanking ventral mesoderm into lateral and dorsal mesoderm, thereby determining the anterior-posterior body axis. And they called this phenomena as embryonic induction, which is now called the primary embryonic induction. And they called this region as the primary inducer or the neural inducer. Actually, the concept of primary embryonic induction, the secondary and the tertiary embryonic induction came quite late after the experiment done by Spemann and Mangold, in which they brought out this concept of organizer. Now it is known that embryonic induction is not a single step, but it is a cascade of events in which there are certain primary induction, secondary induction, and tertiary inductions. Further, some experiments were done by Peter Newcoop and Osama Nakamura. They brought in the concept of Newcoop center and also helped understand how the organizer is formed. The experiment done in 1970 by Nakamura and Takasaki showed that mesoderm arises from the marginal or the equatorial cells, which are present at the border between the animal capsules and the vegetal cells. The animal capsules gave formation of the ectoderm, that is, they are the prospective ectodermal cells. The vegetal cells are the prospective endodermal cells, and thus the marginal equatorial cells are the prospective mesodermal cells. And the properties of this newly formed mesoderm can be induced by the vegetal cells, which are the presumptive endodermal cells underlying them. Another experiment done by Newt showed that if the mesoderm is removed, the animal cells and the vegetal cells will continue forming their respective organs if they are not brought into any kind of contact with each other. But if the animal cap cells are brought into direct contact with the vegetal cells, then the animal cap cells can be induced by the vegetal endodermal cells to form mesodermal structures, that is the notochord, muscles, kidney cells, and blood cells. They also found that whether these induced animal cap cells will form dorsal mesoderm or ventral mesoderm depended upon where the endodermal signals were coming from. The ventral or the lateral vegetal cells, means which are closer to the sperm entry point, induced the formation of ventral and intermediate mesoderm, while the dorsalmost vegetal cells specified the dorsal mesodermal components, the somites and the notochord, plus the cells having the properties of the organizer, that is the dorsal blastopore lip. Thus it was concluded that the vegetal cells underlying the prospective dorsal blastopore region are actually responsible for initiating gastrulation, or that the organizer or the dorsal blastopore lip receives its special properties from signals coming from the prospective endodermal or the vegetal cells beneath it. And these prospective endodermal or the vegetal cells were named the Newt center. The Newt center is actually present in the dorsalmost vegetal region of the xenopus blastula. This Newt center sends the dorsal mesoderm inducing signals, which gives rise to the primary organizer or the Spemann organizer, which is the dorsal lip of blastopore in the xenopus blastula. Now this primary the Spemann organizer further has a dorsalizing effect. It together with the sperm entry point gave rise to the dorsoventral axis. So here we have the concept of Newt center as well as the Spemann organizer. This was further confirmed by some other transplantation recombination experiments done on xenopus embryos by Gimlich and Gerhard, Dale and Slack and Holowash and Ellenson. Now, what gives the dorsalmost vegetal cells their special properties? Or what is the molecular basis of Speman's organizer? The first and the foremost molecule is the beta-catenin, also known as the dorsal signal. Beta-catenin is initially synthesized throughout the embryo from the maternal mRNA stored in it. However, later it is accumulated in the dorsal region of the embryo during the cytoplasmic movements that occur during fertilization and early cleavage. Here, there is an important role of other proteins also, like WNT11, GSK3 binding protein or the GBP, disheveled protein, TCF3, SMAD2 and several genes like SYAMOA and TWIN. The second important factor is the vegetal nodal related signal. Here, VEG-T and VG1 are the vegetal localized proteins which are encoded again by the maternal mRNA stored in the vegetal cells. They encode certain nodal paracrine factors, and they are required for the endodermal formation of the vegetal cells. And further, the vegetal cells will instruct the cell layers lying above them to become mesoderm. The formation of the organizer can better be understood with the help of this flowchart. During cortical rotation, there is translocation of disheveled protein and WNT protein toward the dorsal side of the embryo, where they bind to GSK3. This helps in accumulation of beta-catenin on the dorsal side of the embryo. Now, this beta-catenin during the process of cleavage enters the nucleus where it binds to TCF3 and thereby activates the genes SYAMOA and TWIN, thereby producing the SYAMOA and TWIN transcription factors. On the other hand, beta-catenin also binds to VEG-T proteins in the Newt center, that is the dorsal most vegetal region of the embryo, whereby it activates the TGF-beta family, that is the nodal related proteins VT1 and activin. This helps in phosphorylating and thereby activating the SMAD2 transcription factor. Now, both these pathways ultimately help in activating the organizer genes, that is the cordon, nodin, and goosequoit proteins. So ultimately, it is the intersecting pathways of both sides which help in activating the organizer and help in the formation of the dorsal mesoderm. The first pathway we can say is the WNT beta-catenin pathway, which activates genes encoding the SYAMWA and twin transcription factors. The second pathway is the vegetal pathway that activates the expression of nodal related paracrine factors, which in turn activates the SMAD2 transcription factor in the mesodermal cells above them. The high levels of SMAD2 and the SYAMWA twin transcription factors work together in the dorsal mesodermal cells and activate the genes that give these cells their organizer properties. The work of SMAD2 transcription factor can better be understood with this diagram. This diagram we discussed during gastrulation of the frog also. So here SMAD2 binds to HHEX promoter and in concert with twin and SYAMWA induced by beta-catenin specifies the pharyngeal endodermal cells to become foregut endoderm, thereby inducing anterior brain development. Slightly lower levels of SMAD2 activate the goosequoit expression in prospective precaudal mesoderm and notocaudal cells. Even lower amounts of SMAD2 result in the formation of lateral and ventral mesoderm. So the formation of Speman's organizer occurs through nucleus center, and this formation happens in two different stages as has been studied in the amphibian Xenopus levis. The first stage is the cortical rotation of the fertilized egg. That is, during the formation of grey crescent toward the dorsal side of the embryo that lays the foundation of the dorsoventral axis, there is localization of determinants to the future dorsal side of the embryo that leads to the formation of nucleus center in the dorsalmost vegetal cells. These nucleus center cells remain endodermal in nature. They release certain signals which induce the formation of Speman's organizer in the region immediately overlaying it, that is the equatorial region. And the Speman's organizer becomes dorsal mesoderm. It contributes to the four cell types: the pharyngeal endoderm, the head mesoderm or the precaudal plate, the dorsal mesoderm, primarily the notochord, and the dorsal blastopore lip region. So what functions the organizer has in the formation of embryo? First is the ability of the organizer to self-differentiate into dorsal mesoderm, that is, the formation of precaudal plate and cauda mesoderm. Second, it has the power to induce its surrounding tissues. It dorsalizes the surrounding mesoderm to form paraxial mesoderm or the somite-forming mesoderm. It dorsalizes the ectoderm to form the neural tube, and it initiates the movements of gastrulation in the embryo. There is also seen some regional specificity in the gastrulation. The anterior part of the archenteron roof induces head formation and is called the cephalic or the head inductor. This head inductor further is achencephalic, the portion which induces forebrain, and deuterancephalic, the portion which induces hindbrain. The posterior inducing region is known as the trunk or the spinocaudal inductor. There have also been certain experiments in other organisms to show the primary organizer regions. For example, the experiment done by W.L.Wadington to show the primary organizer in birds. In this experiment, Henson's node was inserted between the epiblast and hypoblast of chick embryo. It led to the induction of a secondary embryo, similar to the experiment done by Spemann and Mangold in newt embryos. So, Henson's node was considered equivalent to the dorsal blastopore lip of the amphibian gastrula, or the Henson's node was considered the primary organizer in chick embryo. However, the posterior one-third of the primitive stray could not induce neural differentiation. That means it did not have any inducing capacity. So it was considered equivalent to the latero-ventral lips of the blastopore of amphibian gastrula. There are various types of embryonic induction. The first is the endogenous versus exogenous induction. Endogenous induction is the one in which inductors are produced by the embryonic cells themselves by the process of self-transformation or self-differentiation. For example, endogenous induction is seen during the formation of dorsal lip of amphibian blastopore. Exogenous induction is the one in which some external agent or cell or tissue introduced into an embryo exert their influence on the embryo through contact induction to form an inducer tissue. Now, this exogenous induction can be homotypic or heterotypic in nature. Homotypic when the inductor provokes the formation of same type of tissue. Heterotypic when the inductor provokes the formation of a different type of tissue. For example, formation of secondary embryonic axis by an implanted presumptive notochord in case of amphibians. Now let us understand the concept of secondary and tertiary organizers. There is not a single step of embryonic induction, but it is actually a cascade of events occurring. Thus, there is a sequential inductive interactions going on which help in the formation of a complete organ. And this interaction process starts with the primary organizer. For example, in the development of Xenopus I, the cells present in the argenteron roof act as primary organizer. They induce the formation of optic vesicles in the brain. Further, the optic vesicles act as secondary organizer and they induce the formation of lens in the head ectoderm. This head ectoderm or the lens region further acts as the tertiary organizer and it induces the formation of cornea. Further, cornea also induces the optic vesicles to form optic cup, and this is a case of reciprocal induction. So the formation of Xenopus I involves the primary organizer, the secondary organizer, a tertiary organizer, as well as a reciprocal induction phenomenon. Then is the concept of instructive and permissive interactions brought forward by Howard Holzer in 1968. In instructive interaction, a signal from the inducing cell is absolutely necessary for initiating new gene expression in the responding cell. In the absence of a signal, the responding cell is not able to differentiate that way. For example, in case of Xenopus optic vesicle experiment, when the optic vesicles are placed under a new region of head ectoderm, they induce it to form lens. And this induction happens by the production and secretion of certain paracrine factors by the optic vesicles. The permissive interactions are the ones in which the responding tissue has already been specified. It just needs a suitable environment to be able to be expressed. For example, many tissues need only the extracellular matrix and the proteins like fibronectin, collagen, and laminin to develop, though they already have been specified to form the tissue further. So with the discussion of the embryonic induction and competence, let us discuss the lecture in a snapshot. Embryonic induction is the induction of embryonic cells to differentiate into organs in the presence of other cells. Inducer or inductor is the embryonic tissue which exerts the inductive effect by producing paracrine factors as signals and secreting them into extracellular space so as to induce the neighboring cells. Responder is the cell or tissue which responds to inductive effect of the inducer, and competence is the ability of responder to respond to inductive effect of the inducer. Speman's demonstration of nuclear equivalence in newt Triturus taeniatus cleavage concluded that early amphibian blastomeres are genetically identical, they have identical nuclei, and each cell is capable of giving rise to an entire organism. Spemann also demonstrated asymmetry in the amphibian egg and concluded that the grey crescent area contains cells committed to the initiation of gastrulation. Another experiment done by Spemann using differently pigmented newt embryos demonstrated that the grey crescent area contains cells committed to the initiation of gastrulation. Transplantation experiments done by Hans Spemann and his doctoral student Hild Mangold demonstrated the organization of a secondary axis by dorsal blastopore lip tissue, and also concluded that the dorsal blastopore lip forms the Speman's organizer tissue of the amphibian gastrula. This tissue dorsalizes the ectoderm, transforming it into neural tissue, and also ventral mesoderm into lateral and dorsal mesoderm. The organizer consists of pharyngeal endoderm, head mesoderm, notochord, and dorsal blastopore lip tissues. The dorsal ventral specification begins with maternal mRNA and proteins stored in the vegetal cytoplasm. These include transcription factors such as VEG-T and nodal-like paracrine factors. And the organizer is induced itself by the nucleus center located in the dorsalmost vegetal cells formed by accumulation of beta-catenin. Experiments in birds confirmed Henson's node to be the primary organizer in case of birds. Sequential inductive interactions occur to form a complete organ during organogenesis. This involves primary, secondary, and tertiary organizers, as seen in the case of development of Xenopus I. There also occur reciprocal interactions during inductive interactions, and we also discuss the instructive and permissive interactions. So now students, try to think upon certain questions on the basis of your understanding of this lecture. Can you predict the effect of blocking cortical rotation in fertilized Xenopus egg upon gastrulation? Think in terms of embryonic induction also. What will happen if the dorsal lip of blastopore from an early Xenopus gastrula is grafted onto the ventral side of an early embryo? You can take help from the various experiments that we discussed during this lecture to answer this question. If beta-catenin is experimentally removed from an early Xenopus gastrula, what will happen? Since we discussed that beta-catenin is an important factor in embryonic induction, so you can think of an answer to this question. Can you explain the role of various maternal factors packaged into the vegetal region of the Xenopus oocyte upon gastrulation? So you have to club all the maternal factors that we discussed in this lecture. How does the nucleus center induce the dorsal organizer? Also, try to find out the role of microtubules in intracytoplasmic movement of proteins during the formation of Speman's organizer in Xenopus embryo. You can find answers to all these questions in the references given at the end of this lecture. Now we'll begin with the second part of this lecture, that is the fate of germ layers, the ectoderm, mesoderm, and endoderm. The fertilized egg or the zygote develops to form a blastula. The blastula further develops the three germ layers to form a gastrula. Now the three germ layers, the ectoderm or the outer layer, the mesoderm or the middle layer, and the endoderm or the internal layer have to undergo organogenesis to form different organs of the body. The ectoderm or the outer layer will form the outer surface, which will form the epidermal cells of the skin, the central nervous system, also known as neural tube, and the neural crest. The mesoderm is further divided into 5 parts: the dorsal, paraxial, intermediate, lateral, head. The endoderm forms the digestive tube, pharynx, respiratory system, while the germ cells are differentiated early in the development, so they form altogether differently. So one by one, we will discuss the fate of the three germ layers here. The ectoderm or the outer layer forms the outer surface of the body, the central nervous system, and the neural crest. The outer surface forms the epidermis of the body of the organism and also various derivatives of the epidermis, which includes the scale, nail, hair, the sebaceous and mammary glands, the internal ear, the lens, conjunctiva, cornea, the olfactory epithelium, as well as the mouth epithelium. The central nervous system develops into the neural tube, which includes the brain at the anteriormost part of the body, along with it the spinal cord, which runs behind the brain till the caudal end of the body. It also develops the cranial nerves as well as the spinal nerves, beside developing into retina, pituitary, and pineal body. The neural crest develops the peripheral nervous system, the adrenal gland, the medulla, as well as certain cells, melanocytes, Schwann cells. It also develops into branchial skeleton, facial cartilage, and dentine of teeth. The function of gastrulation is to place mesoderm in between the ectoderm and the endoderm. Now, the mesodermal regions and their derivatives may be summarized into 5 different parts: the dorsal, paraxial, intermediate, lateral, and head. The regions of the trunk and head mesoderm and their derivatives may be summarized better in this illustration as shown here. The central region of trunk mesoderm is the cauda mesoderm, also known as the axial mesoderm. This tissue forms the notochord, a transient tissue whose major function is inducing and patterning the neural tube under which it lies, and also establishing the anterior-posterior axis of the body. The notochord extends beneath the neural tube from the posterior region of forebrain, and it extends up to the tail. On either side of neural tube further lie thick bands of mesodermal cells, as you can notice here, which are classified as paraxial mesoderm, then intermediate mesoderm, and the farthest are labeled the lateral plate mesoderm. Now, tissues which develop from the paraxial mesoderm will be located in the dorsal side of embryo on either side of the spinal cord. Now, these tissues will form somites. What are somites? The transitory epithelial blocks of mesodermal cells which are found on either side of the neural tube. And these somites give rise to deeper parts of the skin, that is the cartilage, the tendons, the skeletal muscle, the dorsal aorta, which is formed of the endothelial cells, as well as the dermal and skeletal muscles of the body. The intermediate mesoderm is positioned lateral to the paraxial mesoderm, and this intermediate mesoderm forms the urinogenital system, that is the embryonic kidneys, the gonads, and also the associated ducts. Plus, they also form the outer or the cortical portion of the adrenal gland. Now, farthest away from the neural tube are lying the lateral plate mesoderm. There are 2 segments of lateral plate mesoderm: the splanchnic mesoderm, which gives rise to the circulatory system of the body, that is the heart, blood vessels, and blood cells, while the somatic mesoderm forms the smooth muscles of the internal organs and the connective tissue of the pelvic and limb skeleton, except the limb bones, which are somatic in origin. Now, the lateral plate mesoderm also helps in the formation of extra-embryonic membranes. However, the anteriormost paraxial mesoderm does not segment, and it becomes the head mesoderm in the head region, where it gives rise to the connective tissues and the muscles of head. The endoderm, or the internalmost layer of the gastrula, give rise to the digestive tube as well as the associated organs. It forms the lining of the archenteron and associated glands like the liver, pancreas, and gallbladder are endodermally derived. It also forms the pharynx and the associated glands like thyroid, parathyroid, and thymus. Endoderm further also develops into the respiratory system. It forms the complete respiratory tube. It also forms the lung cells as well as the eustachian tube. So summarizing the fate of the three germ layers, the ectoderm generates the outer layer of the embryo. It produces the surface layer or the epidermis of the skin and also forms the brain and nervous system. The mesoderm becomes sandwiched between the ectoderm and the endoderm. It generates the blood, heart, kidney, gonads, bones, muscles, and connective tissues. The endoderm becomes the innermost layer of the embryo and produces the epithelium of the digestive tube and its associated organs, including the lungs. In both the lectures, embryonic induction and organizers, as well as the fate of germ layers, the following books were consulted. Hope you had a good time learning with me. Thank you.